Dr. Zhao is a research scientist and deputy director with BGI Institute of Applied Agriculture, Shenzhen, and he has a great interest in plant evolution, crop domestication, and crop breeding. With this short introduction, I request Dr. Zhao to start his presentation. Thank you.
Thank you for your introduction. Before the presentation, I would like to appreciate the organizers' invitation, invite me here. It's also my honor. As you know, for the past years, BGI collaborates with Dr. Rajim Washley on lots of sequencing projects, from the BGP to chickpea and to other beans and peas. So most of these projects ended up with high-profile publications. So, and during these collaborations, BGI and Dr. Rajim has built a close relationship, and we have lots of fond memories. So today, on behalf of BGI, I bring a small gift to Dr. Washley to salute our friends. See, this is a small disc. Guess what's in it? Yeah, right. His personal genome data. So would you please?
Shancen, thank you very much, and please convey my greetings to Professor Huang Wengyang and other BGI team member to do this thing. So I'm not sure, so till recently, people were having doubt whether I'm a good guy or bad guy, but I'm not sure that how this is going to prove. So I hope that this should have the good genes. Thank you.
I didn't see his data. I remained it to him for his analysis. I think is better. Okay. Today I will. Okay. Today I will talk something about breeding and nutrition. Why choose this topic? It's not easy. You know, BGI has collected a lot of sequencing data on crops, and we also accumulated lots of sequencing data, clinical sequencing data from different hospitals and researches for humans. So I believe that there will be a bridge between the crop genomics and the clinical genomics. So the answer, I think, it will be the precision nutrition. But it's not easy to talk with this, and I also feel a little pressure that I know lots of distinguished professors are sitting here, and of them, including my PhD supervisor, is also here. Today, my talk will include these 4 sections. First, I will briefly introduce the advances of plant genomes in recent years. As you know, every organism has a genome from the prokaryotes to eukaryotes, but their genome size and complexity is quite different. And to decode these genomes, we have lots of sequencing platforms that are available in the markets, from the, as you're familiar with, the Illumina to BioNano and to PacBio. And especially this during the past 3 years, the long-read sequencing technologies has made a great achievement. So at present, the sequencing of long reads is much cheaper and much faster than previously. So based on these achievements, I think that the genome reference projects steps into its gold version. I mean that most of the draft genomes published several years ago, that's just based on NGS sequencing reads, the short reads. The quality of the draft genomes ways to be updated, especially after the new long-rate sequencing has been achieved. So we initialized several genome update projects, including the rice, maize, apples, and some other we have published before. We integrate the different rays from the NGS rays to the BacBio rays and BioNano and 10x genomics. Integrate these different sequencing rays together, we could achieve we could update the genome from the megabase level to chromosome level and to even nearly complete genome size for some simple genomes. I mean, here is a typical procedure to achieve a better assembly. We began from the Illumina rays and PacBio rays to construct a connected hybrid contiguous, and then we added the information to link the different contiguous together to build large-scale scaffolds, and when we used the PacBio data and 10x data, PacBio data, we can construct the contiguous to some chromosome level. So the genome assembly is quite better now. Recently, you know, there are lots of plant genomes that are published in different journals. Some of them are published in very high-profile journals, including Nature, Science, like that. Based on the genome assembly quality and its complexity, and of course its importance in breeding and in applications. But it's getting harder to publish the genome paper in high-profile journals. And here is figure shows that when using different platforms, you will achieve different standards of the genome assembly. You see, recently the PacBio data will help improve the length of contiguous and scaffolds quite a lot compared with NGS data. But unluckily that most of the genomes published at current stage are just based on NGS data. So that's why we want to initialize the genome update project with integrating with different long-range sequencing. Since then, I think BGI has published more than 200 plant species. Some projects are ongoing under review process by BGI. From this figure, I think that most of the contribution was made by monocultures, and I'm also familiar with legumes because my PhD thesis is on this. And I think Dr. Rajim is also familiar with these legumes too. But this genomes, most of the genomes I showed here, ways to be improved by new technologies. But with this genome sequencing data, we could do a lot of things, including decipher the mystery of crop origins, as what has been talked on rice by Professor Han Bin. And also, we could elucidate the primary regions of diversity of major crops. Besides that, we use the population genetics as tools, and we also want to have some hot points of agricultural genomics to develop markers, to develop genetic maps and identify genes. And we also use this genome sequence to apply them in breeding through marker development and something like that. 2 years ago, I wrote a review paper together with our collaborators to summarize the change of sequencing towards breeding. As you see here, this shows the accumulation of sequencing of knowledge, of breeding knowledge and research results. And here, this shows the sequencing skills. As you see, rice is leading in the first position because the genome size is small, and lots of researchers paid a lot of efforts on this research. And it followed by maize. Amazing you see is its weight. So we accumulate a lot of knowledge on weight breeding and weight researches, but its genome sequencing lags. So recently, this year, a lot of genome, weight genome has been will be published and updated. With the sequencing technology promotes, you see these figures are moving fast towards these sequencing skills. I mean that lots of plant species are sequencing to a large scale. To help the application of genome sequencing to breeding practice, I think the key is to identify useful markers. So that's what we show in this figure. And next, I want to introduce the precision nutrition. Traditionally, there is one solution that fits all in the nutrition and medicine. I mean that when applied screening, diagnosis, medicine treatment, we didn't distinguish the different populations, let alone different individuals. We almost used one solution that fits all, but this model practice now faces many challenges. As the research shows that high variability exists in response to the identical meals in different populations. It suggests that universal dietary recommendations may have limited utility when dealing with different populations. And I believe that the personalized nutrition plate will contain these different aspects like dietary habits, microbiota, metabolism, food habit. And for me, as a consumer, I think there is a customized journey towards the precision nutrition I will explain. For eat and drink, I need to eat healthy and tasty food that's tailored to my needs. I mean, it's personalized to eat. And work. Okay. And for food intake, I want to monitor what I eat quickly and easily, especially in a non-intrusive way. And I also want to measure my health status easily in a non-intrusive manner. So also, when I'm getting a lot of data, I want to make sure this data can be and its relevant information can be modeled to develop some models to give some suitable suggestions on my diary. And I will also need to choose and we need connected coaching and different situations. I mean, we need some recommendations on how to eat and how to choose the dietary. And we also want to be empowered to adapt my lifestyle to feel good. And we want to find products and services that fit our personalized needs. So that's the personalized, that's a customized view of that towards the precision nutrition. And in fact, it's difficult to define precision nutrition, but there are some goals exist because I think the ultimate goal is to enhance the health and the well-being of society by empowering customers for their personalized needs. And to achieve these goals, I think we need to make more precision measurement of the dietary intakes and more precise diagnosis of nutritional status. And we also want to some personalized nutrition therapy or dietary recommendations based on what the data collected. And in fact, I think there will be a way to solve this. The genetic testing will be an important tool to identify to learn how the genes can affect these different aspects like food tolerance, weight management. Because after a lot of data, I think there will be answers in the genes if we understand it deeper and better. And last, I want to show some brief introduction of our efforts towards this hard task. BGI this year is founded together with the government, founded a new institute of applied agriculture. I am responsible for this, but this new founded institute will focus on four aspects. Of them, precision nutrition will be our strategic importance to do breeding. And I think there will some key phases towards precision nutrition. The first is to build the initial database. So at this stage, we still are collecting different data on different populations. We want to identify, we want to answer who are you by your sequencing data. And the second stage, we want to measure and monitor the different dietary habits. We want to answer how are you during the dietary habits. And we also want to interpret and recommend from this data information to different personalized needs. And we also want to intervene by breeding some certain crops to achieve some personalized to some functional foods products. And based on that, we could provide what should you do, give some certain suggestions. And finally, I want to know how do you feel after this. The first project we initialized is the Chinese Health and Disease Consortium. From this project, we collect the dietary intake and genomic data in a group of adult Chinese, especially the population of diabetes adults. We also collect this data from BGI's employees. And from this different data, at one side we collect we analyze the different components in foods, and we also collect the different data from different persons. We want to link the food consumption with their psychological parameters. We now at the current stage we have accumulated about 3,000 precision data or diagnosis data from the BGI's employees. And we are analyzing the different dietary and these psychological changes. And based on this, we construct a database that including variations related with nutrient metabolism. Here are some famous markers related with genes that will conduct some nutrition-oriented guidance. But we also want to use the breeding as a tool to help improve the nutrition status, because for cancer, we usually didn't have approaches to deal with to cure it, but we will release with some different food treatment. We could release the deleterious status in the hospital. So what we want to develop different foods for different functional foods for these different healthy people, something like that. But the tool can be used is the ideal breeding equation. We have to collect the different multiple omics data, and we have to consider different environments. And this is familiar by most of you, is that we use the genome selection and the pyramiding breeding to accumulate the desirable alleles. Some cases are here. This is a case of foxtail millet breeding. We want to use this to develop some food for some diabetes. And first, we identify the sessions that have robust resistance to drought and other environments. And we also these sessions have high yield. So by combining them together, we developed a new cultivar that have better resistance and higher yield. And after that, we identified a session with low starch. And by crossing them together, we identify some markers that are related with low starch, and then we developed a new cultivar that I think that will be better, that be good for diabetes. And we also have a case of melon for high nutrition. We have one accession that is white fruit but big size but low fructose. And we also have a accession that with yellow fruit but small size is high fructose. If we want to achieve high fructose, we combine these different accessions together. And after round of genome selection, we achieved yellow fruit and big size and high fructose. That will be better in the perspective of nutrition. And besides that, we also initialized some large-scale projects like medicinal plant genome sequencing, and we want to sequence more than 600 plant genomes and their transcriptomes. And based on this data, we want to develop different products that towards precision nutrition. And the other side is we also initialized a large-scale fungi germplasm sequencing project. Why choose this medical plant and fungi germplasm? Because lots of secondary biomolites exist in these different species that will be good for the nutrition. And here is a case on tea plant. We understand, we research the regulation and biosynthesis of catechin pathway, and from this some key genes in the catchway, we could adjust the final components of different catechins to make different teas for different needs. And we also studied the pathways of anthocyanin and proanthocyanidins with our collaborators. Notice that this Zhao is not me, it's our collaborators. And I think this conserved pathway that ended up with different end product will help us in breeding that towards nutrition purpose. We are still doing this. And even with this data, with different tools, I think there is still a long way to go towards precision nutrition. At the current stage, I think most of the relationships are association relationship. We underlying this association relationship, we want to identify which factors are the causal ones that determine the different components and the human psychological factors. So I think we are still at the stage of collecting different data, and after analyzing these data, we want to connect the different associations between the factors and identify the true causal factors that help us make decisions. But besides this data, I think the gut microbiota will add another layer of complexity. You know, there are lots of genes, lots of microbiota in human gut. So this is also our important focus that towards nutrition guidance. So we also initialized a lot of gut microbiota project together with BGI research and other institutes. And finally, we want to construct a database, a supporting system that help data-driven healthcare and give some nutrition guidance. This system is hopefully will be online next year, and together with the knowledge, the current knowledge, biological knowledge, the psychological knowledge, and the personal goals, we want to finally help different people make decisions that towards nutrition, towards personalized nutrition. So that's our goal, and we are still doing. And I think there is it's not easy, a long way to go. Here is a new institute that located in Shenzhen, and this is my office located in this building. So thank you for your attention. Thank you all.
Thank you, Dr. Zhao, for a nice presentation, and also letting us know that where this sequencing is moving. Going from probably means we see that next 5 years probably we will be moving towards the sequence-based.
Yeah, right.
nutrition. Thank you very much.
Thank you.
Time
Any questions?
for questions.
Yes, please.
Short question. You mentioned the gut microbiome, that you sequenced the gut microbiome. You're also interested in the plant microbiome?
Yeah, in fact, BGI has made a great contribution in gut microbiome research. The diversity of microbiome species is much more than the human genes in the gut. Generally, there will be 3 kilobase microbiota in human gut, and there will be about 3 million genes there. So it's really complex, and we treat at this stage we focus on some pregnant women and diabetes to research the microbiome. Yeah, plant. In fact, we just published a paper to elucidate the plant microbiota on fox millet. Yeah, it's really helpful in the plant nutrition. Yeah.
So I have one technical question. So you presented this long reads are effectively used for assembling the genomes.
Yeah.
So my question is, I want to use these long reads to map against the existing reference genome. So which tool you suggest to use that to map the long reads to the reference genome? Because all the existing tools are basically using the small reads.
In fact, I'm not sure I catch your question.
What is the best tool to map against or align against the reference genome for these long reads?
I think it's lies in how to explain, how to apply the knowledge. At the current stage, we can test a lot of genes, a lot of loci, but our understanding on this are still far, I think, to give precision decisions on this.
One more question.
Okay.
Last one. Yeah. Okay.
Can I have the quick question? Have you worked together with someone, your neighbors, Agricultural Institute, Genomic Institute, because they are doing similar, the work sequencing the, from the genome.
In fact, several years ago we collaborated a lot, but after that our collaboration getting weak. And from this year we also strengthen our collaborations on cucumber. The genome of cucumber was finished by our group together, and I also discussed these goals and this research direction with Professor Huang a lot, and also I get a lot of information from him. But I will not focus on cucumber. We just focused on general species. Yeah. Yes. Thank you.
Okay. Let's move on. Thank you, Dr.
Thank you.